The human brain uses roughly 20 watts of power to run. 20 watts. That is less electricity than a dim bedside lamp. Meanwhile, the most powerful supercomputer ever built burns through tens of megawatts just to keep its processors from melting. The energy gap between the brain and its closest silicon rival is approximately 1 million to 1. If the brain was simply doing the same kind of computation that a computer chip performs, that ratio would be physically impossible. You cannot run the same class of calculations on 1/1,000,000th of the energy. The thermodynamics collapse. So either we fundamentally misunderstand what computation is, or the brain is doing something entirely different from what our machines are designed to do. And the evidence, increasingly, clearly, and uncomfortably, points to option 2. This is not a gap that more transistors will close. It is not a problem waiting on a faster chip or a more efficient cooling tower. It is a problem rooted in physics itself. The brain and the classical computer are operating according to different physical rules. And until we build machines that follow the same physical rules as biology, a true simulation of the mind will remain out of reach. That situation is beginning to change in ways that nobody predicted a decade ago. To understand what is happening, we first need to appreciate the scale of what we are actually trying to copy. Most people dramatically underestimate it. The brain contains roughly 86 billion neurons. That number sounds enormous, and it is. But the neurons themselves are not where the real complexity lives. The complexity lives in the connections between them. Each neuron is linked to thousands of others through microscopic structures called synapses. When you map the complete network of connections in a brain, the structure that researchers call the connectome, you arrive at approximately 100 trillion distinct synaptic connection points. 100 trillion. To hold that number in perspective, there are roughly 10 times more synaptic connections in a single human brain than there are stars in the entire Milky Way galaxy. And these connections are not static. They are not a fixed wiring diagram printed once and left alone. Every second, hundreds of trillions of synapses are either strengthening or weakening in response to incoming chemical signals. The brain is a living, self-reconfiguring network that never sits still. Simulating that level of dynamic, massively parallel activity on a machine that processes information sequentially is like trying to capture an ocean using a series of still photographs. You can approximate it. You will always miss the motion. The most ambitious attempt to tackle this problem directly using classical hardware was the Blue Brain project, launched in 2005. The goal was to build a biologically detailed digital reconstruction of the mammalian brain from the ground up. Starting with the basic unit and scaling outward, the team used some of the most powerful supercomputers available at the time. After years of sustained effort, they succeeded in simulating a small neocortical column taken from a rat brain, a sliver of tissue containing roughly 30,000 neurons. To run that simulation, the researchers had to track every ion channel, every electrical spike, and every chemical gradient across all 30,000 cells simultaneously. The computational cost was staggering, and that was for 30,000 neurons. A human brain contains 86 billion. When researchers tried to project what it would take to scale that simulation up to a full human brain at the same level of biological detail, the numbers became effectively unreachable. They would need computational resources on the scale of a large industrial facility just to store the data, let alone process it in anything approaching real time. The classical approach had hit a wall that no amount of chip fabrication could dissolve. The problem became vivid in 2013. A team used Japan's K supercomputer, one of the most powerful machines on the planet at the time, to simulate just 1 second of brain activity. The simulation represented only about 1% of the full human brain. After the model was loaded and the calculation began, the K computer needed 40 minutes of processing time to generate that single second of neural activity. 40 minutes of supercomputer time to recreate 1 second of biological thought. That is not a simulation. That is an extraordinarily slow-motion replay of a system running thousands of times faster than the computer trying to capture it. There is no path from that ratio to real-time simulation. By piling on more classical hardware, more recent work has made genuine progress. In 2025, a team used the Fugaku supercomputer, which succeeded the K computer as Japan's flagship machine, to simulate the mouse cortex with nearly 10 million neurons and 26 billion synapses. They achieved performance approaching real time. This was a significant milestone, but it required simplifying the biological model substantially and running it on hardware that costs billions of dollars to build. And the mouse cortex is a fraction of the complexity of a human brain. The gains in classical computing have been real. The ceiling is still not high enough. What makes this particularly difficult is not just the neuron count. It is the architecture. A classical computer is fundamentally a linear machine. Data moves from storage to processor, gets calculated, and moves back. Even with parallelism, the underlying structure is flat. The brain is not flat. It is a three-dimensional, fully interconnected mesh where every part of the network can affect every other part simultaneously. Running a three-dimensional, interconnected biological network on a two-dimensional sequential architecture is the source of the bottleneck. We have reached the edges of what silicon can do. Now, consider the energy problem, because it reveals something much deeper than just efficiency. When you look at a modern supercomputer facility, most of what you are looking at is not computing, it is cooling. These machines generate so much waste heat that they require industrial cooling towers and a continuous supply of water, just to keep the processors from destroying themselves. The actual computation is a fraction of the total power budget. Most of the energy goes into fighting heat. The brain generates almost no waste heat. If you put your hand near a thinking human brain, you would not feel warmth radiating off it the way you feel from a running processor. The brain solves complex problems, generates language, processes vision, coordinates movement, manages emotion, and runs the machinery of consciousness on a metabolic budget that your body sustains through a modest diet. 20 watts continuously for decades. This discrepancy is not just an engineering curiosity. It is a physical clue pointing toward a different kind of computation entirely. In classical computing, heat is a fundamental byproduct of making decisions. When a processor switches a transistor from 0 to 1, it is physically forcing electrons through a resistive gate. That physical act generates friction, and friction generates heat. The more decisions you make, the more heat you produce. This relationship between computation and thermodynamics was formalized by physicist Rolf Landauer in 1961. Landauer's principle states that erasing a single bit of information requires a minimum amount of energy, and that energy must be released as heat. Every time a computer discards a possibility to reach a conclusion, it pays a thermodynamic price. You cannot get around this in classical computing. It is baked into the physics. But the brain does not seem to pay this price in the same way. It is not overcoming resistance to force binary decisions. It is computing in a way that bypasses the Landauer limit, or at least skirts it dramatically. One reason this might be possible is that quantum computation can, in theory, be reversible. A reversible computation leaves no information behind. It erases nothing, and if nothing is erased, no heat is generated. The brain does not achieve perfect reversibility, but its efficiency suggests it is operating closer to that regime than any silicon chip ever has. This points toward a different physical mechanism underneath the biology. And the most compelling candidate is quantum mechanics. For a long time, suggesting that the brain might operate on quantum principles was considered fringe science. The standard objection was simple and seemed decisive. Quantum states are extraordinarily fragile. To maintain quantum coherence, a system typically needs to be isolated from its environment, cooled to temperatures near absolute zero, and shielded from any external vibration or electromagnetic interference. The brain is warm, wet, chemically chaotic, and constantly bombarded with thermal noise. The assumption was that any quantum state inside a neuron would collapse within femtoseconds, far too fast to play any role in cognition. This argument seemed airtight. Then scientists started looking more carefully inside the neuron itself. For decades, neurons were understood primarily as signal cables. The outside membrane carried the electrical impulse. The interior was thought to be just plumbing, structural support, nothing interesting. That picture has changed substantially. The interior of a neuron is packed with a dense structural scaffold called microtubules. These are hollow, cylindrical protein structures that form the skeleton of the cell. They are found throughout biology. In most cells, microtubules are pure scaffolding. But inside neurons, they may be doing something far more important. Physicist Roger Penrose and anesthesiologist Stuart Hameroff proposed a theory they called orchestrated objective reduction, or ORC-OR. They argued that microtubules are not merely structural supports. They suggested that the geometry of the protein chains forming the microtubule wall, and the arrangement of electrons within those chains, might create a partially insulated quantum environment inside the tube, if correct. This insulation would allow quantum states to persist long enough inside the neuron to participate in information processing. The microtubule would act as a biological quantum waveguide, shielding coherent quantum states from the thermal noise of the surrounding cellular environment. The scientific community was skeptical of this idea for years, and the skepticism was reasonable. Extraordinary claims require extraordinary evidence, but the evidence has been accumulating in ways that are increasingly difficult to dismiss. Researchers have observed quantum vibrations in the protein lattice of They have found that energy can propagate through these structures with anomalously low loss, behaving more like a coherent wave than a classical thermal disturbance. Studies have linked this propagation to networks of tryptophan molecules inside the tube. When these molecules resonate together, they appear to generate a macroscopic quantum state, similar in principle to how a laser works. In a laser, individual photons synchronize into a single coherent beam through stimulated emission. Inside microtubules, molecular resonances may synchronize into a single coherent quantum state that spans the length of the tube. This is not proof that the brain runs on quantum mechanics, but it is no longer dismissible as fantasy, and it fundamentally changes the conversation about simulation. If there is quantum processing happening inside neurons, even at a limited scale, then a classical computer cannot faithfully replicate it. A binary switch cannot simulate a quantum superposition. These are not just different speeds of the same process. They are different physical categories. A simulation that ignores the quantum layer of the brain is not a simulation of the brain. It is an approximation of certain statistical behaviors that the brain produces, which is a very different thing to understand why quantum computers offer a genuine solution. We need to understand what makes them fundamentally different from the classical machines we know. A classical bit is always either 0 or 1. It has no ambiguity. A quantum bit, called a qubit, can exist in a superposition of 0 and 1 simultaneously. This is not a metaphor or a figure of speech. It is a physical reality. The qubit is genuinely in both states at once until it is measured. When you build a system of multiple qubits, they can also become entangled. Entanglement means that the state of one qubit is instantaneously correlated with the state of another, regardless of physical distance. The two qubits are no longer independent. They are a single, unified quantum system. Measuring one instantly determines the state of the other. This is where the power of quantum computation comes from. When you solve a problem with classical bits, you explore possible solutions one at a time or in parallel batches, always moving through discrete states. When you solve a problem with qubits in superposition, the machine is exploring all possible solutions simultaneously through quantum interference. The probability amplitudes of wrong answers cancel each other out. The probability amplitudes of correct answers reinforce each other. The computation does not guess and check. It converges on the answer through the physics of wave interference. For simulating physical systems like the brain, this matters in a specific and decisive way. To model any physical system, you need to describe its energy state. In physics, this description is called the Hamiltonian. The Hamiltonian is essentially the energy fingerprint of a system, capturing how every component vibrates, interacts, and evolves over time. A single neuron has a Hamiltonian. A network of neurons has an incomprehensibly more complex one. If you can accurately define the Hamiltonian and simulate how it evolves, you have simulated the system. When a classical computer tries to simulate a Hamiltonian, it has to solve the mathematics explicitly, computing the differential equations that describe the evolution of the energy state one step at a time. As the system grows, the equations grow exponentially harder. At the scale of even a small neural circuit, the classical computer essentially freezes. It is trying to describe every ripple in a pond by tracking every water molecule individually. A quantum computer does not try to solve the equation. It tries to become the equation. You configure the qubits so that they are governed by the same physical laws that govern the neurons. You set up the interactions between qubits to mirror the interactions between biological molecules. You are not calculating what the neuron will do. You are building a quantum replica of the neuron and observing what it does. The hardware and the simulation are the same thing. This idea goes back to Richard Feynman, who made it precisely in the early 1980s. He observed that nature is not classical. Nature is quantum. Therefore, if you want to simulate nature, you need a quantum machine. For 40 years, that prescription sat waiting for hardware capable of fulfilling it. The practical bottleneck until recently was error. Qubits are extraordinarily sensitive to their environment. A stray magnetic field, a temperature fluctuation of a fraction of a degree, a cosmic ray passing through the chip, any of these can cause a qubit to flip from its correct state to an incorrect one. These are called quantum errors, and for years they accumulated faster than any algorithm could run. The computers were powerful in theory and unusable in practice. The field of quantum error correction existed to address this, but for most of its history, it faced a cruel paradox. Adding more physical qubits to implement error correction codes also added more potential failure points. The system became noisier as it grew larger. You were fixing errors at the bottom while introducing new ones at the top. This situation changed fundamentally in late 2020, for with the announcement of Google's Willow quantum chip. Willow was not simply a faster quantum processor. It was a demonstration that the error correction paradox could be broken. The team at Google showed that by grouping physical qubits into logical qubits, clusters of physical qubits that collectively protect a single unit of quantum information. The error rate decreased exponentially as they added more physical qubits to each logical group. For the first time, making the system larger made it more stable rather than less. Adding more physical qubits exponentially reduced errors rather than amplifying them. This is the turning point that the field had been waiting for. It is the proof of concept that quantum computers can, in principle, maintain stable quantum states for the duration of a complex computation. For biological simulation, this matters enormously. The brain maintains a continuous state of consciousness for decades. Simulating it requires a computer that can sustain coherent computation not for microseconds, but for minutes and eventually hours. The Willow result suggests that is achievable. We are moving into an era where quantum algorithms can run long enough to observe how a digital neural network evolves and adapts over time. The immediate practical applications of this are in medicine, specifically in the understanding and treatment of neurodegenerative diseases. Diseases like Alzheimer's, Parkinson's, and Huntington's are not simply diseases of neural death. They are, at their molecular foundation, diseases of geometry. The machinery of the nervous system relies on proteins, and proteins must fold into precise three-dimensional structures to function. A protein begins as a long chain of amino acids. To become active, this chain must collapse into a specific shape determined by the interactions between its chemical groups. If it folds correctly, it does its job. If it folds incorrectly, the misfolded protein can become toxic, clumping together into aggregates that damage and kill neurons over decades. This is what drives the progression of Alzheimer's disease. The amyloid plaques that define the disease are the physical residue of misfolded proteins accumulating over years. The challenge of predicting how a protein will fold is one of the most computationally demanding problems in all of science. A modest-sized protein can theoretically adopt more distinct conformational shapes than there are atoms in the observable universe. A classical computer approaches this by guessing a shape, calculating the associated energy, adjusting, and guessing again. This brute force search is so slow that a supercomputer can take months to determine the structure of a single complex protein. The field changed dramatically when DeepMind released Alpha, an artificial intelligence system trained on known protein structures that could predict folding with remarkable accuracy. But AlphaFold is a pattern matching engine. It works by learning from examples. For novel proteins with no structural analogues in the training data, it struggles. And for simulating how misfolding happens dynamically in real time under specific molecular conditions, it is not the right tool. A quantum simulation approaches protein folding from physics rather than pattern matching. In nature, a protein folds by seeking the configuration that minimizes its energy. The chain samples different shapes continuously and gravitates toward the state of lowest energy. A quantum computer can simulate this process directly. The qubits settle into their own state of lowest energy automatically by the physics of quantum mechanics, mirroring the physical process the protein undergoes. The computer does not search through possibilities. It physically enacts the folding. Combine this with the concept of a digital twin, and the implications for medicine become concrete. Imagine scanning the molecular structure of a specific patient's brain, loading that data into a quantum processor tuned to reflect that patient's neurochemistry. You have now created a sandbox replica of that person's neurobiology. You can introduce a candidate drug molecule into the simulation and watch how it interacts with the misfolded proteins in real time. You can observe whether it interrupts the aggregation cascade. You can detect unexpected interactions with other molecular systems before the drug has ever entered a living body. You can run thousands of variants of a compound in a single afternoon. This compresses a development timeline that currently spans over a decade and costs billions of dollars into something measurable in weeks. Beyond medicine, there is an application emerging in the field of brain-computer interfaces, and it involves a problem that has defined and limited the technology since it began: latency. When a person using a neuroprosthetic decides to move a hand, the brain generates a cascade of electrical signals that encode the intention. A classical computer receiving those signals must first filter the noise, then run a decoding algorithm to interpret the pattern, then generate a command, and finally send it to the motor. Each step takes time. The result is a noticeable delay between intention and action. The user does not feel like the prosthetic is part of their body. They feel like they are operating a tool with their thoughts. Neural signals are not clean binary messages. They are probabilistic clouds of electrical activity distributed across many neurons simultaneously, riding on a background of biological noise. A classical decoder must wait until the signal achieves sufficient statistical clarity before it can commit to an interpretation. It discards ambiguity because ambiguity breaks the logic of binary decision. This waiting is where the delay lives. A quantum processor does not need to wait for ambiguity to resolve. A qubit can natively represent a probability distribution rather than forcing a signal into a binary category. The raw, noisy probability field of the neural signal can be mapped directly onto the qubits, which process the entire distribution at once. The quantum system can decode the user's intention before the biological signal has even fully propagated through the neural pathway. This speed is the difference between a prosthetic that requires conscious effort to operate and one that responds as naturally as a biological limb. But the implications extend further than motor control. Motor signals are relatively simple because they encode specific muscle commands with a limited vocabulary. Abstract thoughts are not. When you visualize a face, that visual pattern is distributed across dozens of cortical regions simultaneously. The information is encoded in the correlations between regions, not in any single stream of data. A quantum system can analyze correlations across many brain regions in parallel, detecting the subtle synchronizations that represent specific memories, images, or concepts. This opens the possibility of decoding semantic thought directly, not just motor intention. If the latency is low enough and the decoding accurate enough, the interface stops being an interface. The machine becomes a transparent extension of cognition. To understand what all of this means, philosophically and physically, we need to go back to first principles. In classical mechanics, a system has a definite state at every moment. A billiard ball is at a specific position, moving in a specific direction with a specific energy. If you know the initial conditions precisely, you can predict the entire future trajectory of the system in principle. Reality, in the classical picture, is deterministic and definite. Quantum mechanics replaces this with a different picture. A quantum system is described not by a definite state, but by a wave function. The wave function encodes the probability amplitudes for every possible measurement outcome. When you ask where a particle is, the wave function gives you not an answer, but a probability distribution of possible answers. The particle does not have a definite position before you measure it. It has a superposition of positions. Measurement collapses the superposition to a single outcome. But before measurement, all outcomes coexist. This is not a statement about our ignorance. It is a statement about reality itself. The particle genuinely does not have a definite position before measurement. This was confirmed by a series of experiments testing what are called Bell inequalities. following theoretical work by physicist John Bell in 1964. Bell showed that if quantum particles had definite values of their properties before measurement, hidden from us but objectively real, then certain correlations between measurements on separated particles would have to obey specific mathematical bounds. Experiments beginning with those of Alling aspect in the 1980s and continuing with increasing precision to the present day have consistently violated these bounds. The violations mean the particles genuinely do not have pre-existing definite values. The quantum description is not a shorthand for classical ignorance. It is a description of how nature actually is. For computation, this means something specific. When qubits are placed in superposition, the computation genuinely explores multiple branches simultaneously. This is not parallelism in the sense of having multiple processors each working on one branch. It is the exploration of multiple branches within a single physical system governed by the mathematics of quantum interference. The wrong answers interfere destructively and cancel out. The right answer interferes constructively and emerges. This is fundamentally different from anything a classical system can do, because a classical system can only follow one branch of possibility at a time, even if it has many processors working in parallel. For simulating physical systems, including the brain, this means a quantum computer can evolve the simulation through time using the same equations that govern the actual physics. Quantum simulation is qualitatively different from classical simulation. It is not faster. It is more accurate. It does not approximate the physics. It performs it. The coherence that makes this possible is also what makes it difficult to engineer. Quantum coherence, the property by which different possible states of a quantum system maintain fixed phase relationships and therefore interfere with each other, is destroyed by interaction with the environment. Any coupling between the quantum system and its surroundings causes the superposition to leak information into the environment, and the quantum state collapses toward a classical mixture of possibilities. This process is called decoherence, and it is the central engineering challenge of quantum computing in the laboratory. Decoherence is fought by isolating the quantum processor from its environment as thoroughly as possible. Superconducting qubit chips are cooled to temperatures around 15 millikelvin, colder than the emptiness of outer space, to suppress thermal vibrations. They are shielded from electromagnetic radiation. Control signals are designed to minimize unintended coupling. Even with all these precautions, the coherence time of individual qubits is limited to microseconds, or, in the best systems, milliseconds. In the brain, the environment is exactly the opposite of a carefully isolated quantum laboratory. The temperature is 310 Kelvin, close to thermal equilibrium. The cell is filled with water, which is an aggressive source of decoherence, because water molecules are polar and constantly interact with charged molecules. The cell membrane is subject to constant mechanical vibration, and yet quantum effects in biological systems have been observed. This apparent contradiction, quantum coherence surviving in a warm, wet environment, has become one of the most interesting open questions in biophysics. The resolution may lie in the timescales involved. Decoherence in biological molecules occurs extremely quickly, often in femtoseconds. But the relevant biological processes, such as energy transfer in photosynthesis or conformational changes in ion channels, also occur on femtosecond timescales. The quantum coherence does not need to survive for a long time. It needs to survive long enough to influence the next step in the biological process. If the chemical reaction completes faster than the coherence decays, the quantum effect has done its work. The warm, wet environment that seems hostile to quantum mechanics may be irrelevant if the quantum events are over before the environment can destroy them. This is why the analogy between quantum computers and quantum biology is imperfect but illuminating. A quantum computer is engineered to maintain coherence over millions of gate operations. A biological system uses quantum effects for a single fast step and then allows The strategies are different, but the underlying physics is the same. Both are exploiting the wave nature of matter to accomplish something that would be impossible if reality were purely classical to appreciate. Why quantum mechanics is the right language for the brain. It helps to understand the scale at which biology actually operates. We tend to think of biology as chemistry, and chemistry is something that happens at a level comfortably above quantum physics. That picture is wrong. The molecular machinery inside every cell operates at length scales where quantum effects are not peripheral. They are central. A water molecule is about 3 angstroms across. An angstrom is 1/10,000,000,000th of a meter. The synaptic cleft, the gap across which neurotransmitters travel to transmit signals between neurons, is about 20 nanometers wide. A nanometer is 1/1000,000,000th of a meter. At these dimensions, the behavior of electrons is governed by quantum mechanical wave equations, not by the classical laws that describe billiard balls or planetary motion. The proteins that open and close ion channels in a neuron's membrane are molecular machines operating in this quantum regime. When a voltage-gated sodium channel opens to allow an action potential to propagate, the conformational change in the protein that opens the channel is driven by the quantum mechanical behavior of electrons and atomic nuclei. Classical mechanics cannot accurately describe it. It was never designed to. This means the brain is not a system that happens to be implemented in biology. It is a system whose fundamental operations are quantum mechanical. The neurons and synapses we see at the scale of cellular biology are the emergent high-level output of processes that at their foundation are as quantum as any phenomenon studied in a physics laboratory. Simulating this system classically is not just slow, it is wrong in principle, in the same way that a classical model of hydrogen is wrong. It produces approximately correct answers in some regimes and completely breaks down in others. A classical simulation of the brain can capture the statistical behavior of neural firing rates and average connectivity patterns. What it cannot capture are the quantum correlations, the interference patterns, and the coherent energy transfer that may underlie the brain's most distinctive capabilities, including its efficiency, its flexibility, and possibly its capacity for subjective experience. A classical model of the brain is like a classical model of superconductivity. It works until it fails completely, and when it fails, no refinement within the classical framework can fix it. Feynman's argument about simulation makes this concrete. To simulate a quantum system on a classical computer, you need to track the amplitude of every possible quantum state of the system. For a system of n quantum particles with 2 possible states each, the number of amplitudes you need to track grows as 2 raised to the power of n. For a system of 300 entangled quantum particles, you would need more amplitudes to describe it classically than there are atoms in the observable universe. You cannot fit the description of such a system into any storage medium made of matter. The brain contains vastly more than 300 quantum-relevant degrees of freedom. If even a small fraction of the neural processing involves quantum entanglement and coherence, the classical simulation is not just difficult, it is formally impossible at any scale of hardware that could ever be built. The only machine that can simulate a quantum system of that complexity is another quantum system of comparable complexity. This is Feynman's prescription made concrete. What distinguishes quantum computation from classical? Computation is not the hardware. It is the class of logical operations that the hardware can natively perform. A quantum processor can natively perform operations that are impossible on any classical machine, regardless of how fast the classical machine runs. For brain simulation, the relevant native operations are quantum simulations of physical Hamiltonian. A classical processor cannot perform these operations natively. It can only approximate them through numerical methods that become exponentially expensive as the system size grows. A quantum processor performs them directly. This is not a matter of degree. It is a matter of category now. Consider the connectome and what it means to simulate it properly. The connectome is the complete map of neural connections in a brain. Mapping the connections, knowing which neuron is connected to which other neuron and with what synaptic strength, is only the beginning. The map is static. The brain is dynamic. To simulate the brain, you need to model how activity propagates through that architecture in real time, how synaptic strengths change in response to activity, how neuromodulatory signals globally alter the processing of local circuits, and how all of these processes interact simultaneously across the whole network. The first complete connectome ever mapped at the level of individual neurons and synapses was the connectome of Qubits elegans, a tiny round worm with 302 neurons and approximately 7,000 synapses. This project was completed in the 1980s and took over a decade of manual reconstruction from electron microscopy images. With that complete map, researchers could simulate the worm's nervous system classically, and the simulations were informative. But they also revealed something important. Even with the complete connectivity map, the classical simulation could not fully reproduce the worm's behavior. Something was missing. The map was necessary, but not sufficient. The missing elements are the dynamic properties of the neurons themselves, including their intrinsic excitability, the kinetics of their ion channels, the spatial distribution of inputs across the dendritic tree, and the probabilistic nature of synaptic transmission. All of these properties are quantum mechanical at their foundation. A classical simulation can parameterize them with equations fitted to experimental data. But parameterization is not simulation. It replaces the physics with a model of the physics. And the model is always an approximation. The Human Connectome Project, launched in 2009 with funding from the United States National Institutes of Health, set out to map the large-scale connectivity structure of the human brain using diffusion MRI tractography. This technique traces the pathways of white matter fiber bundles connecting different brain regions. It does not resolve individual neurons and synapses, but it provides a macroscale map of interregional connectivity. These models have generated useful insights, but they cannot simulate the neural activity itself. They model statistical patterns at a spatial and temporal resolution far coarser than the actual neural dynamics. They are maps, not territories. The territory requires knowing what is happening at the synapse, at the ion channel, at the protein level. And at each of those levels, the relevant physics is quantum mechanical. There is also a dimension of this problem that is rarely discussed: the role of glial cells. For most of the history of neuroscience, glial cells were considered to be the support staff of the brain, providing structural scaffolding, supplying nutrients to neurons, and maintaining the ionic environment that allows neurons to fire. They were thought of as passive. Research over the past 2 decades has overturned this picture substantially. Glial cells, particularly the astrocytes, are now understood to actively participate in neural signaling. Astrocytes form intimate contacts with synapses. A single human astrocyte can enwrap as many as 2 million synapses. At each synapse, the astrocyte monitors the chemical environment and releases its own signaling molecules. called gliotransmitters, which modulate synaptic strength. Astrocytes also propagate calcium waves that travel through networks of connected astrocytes at speeds and over distances that are relevant to neural computation. The brain has as many astrocytes as it has neurons. A simulation that ignores astrocytes is ignoring half the computational substrate, including astrocytes, in a brain. Simulation adds a layer of complexity that further highlights the inadequacy of classical approaches. The calcium dynamics inside an astrocyte involve the same quantum mechanical protein behaviors that govern ion channel opening in neurons. The release of gliotransmitters involves the same quantum-level conformational changes in molecular machines. A complete simulation must simulate not just the neurons, but the entire cellular ecosystem of the brain. One area where the near-term payoff is most concrete is in the simulation of anesthetic mechanisms. Anesthetics are among the most widely used drugs in medicine. Yet the mechanism by which they render a patient unconscious is still not fully understood at the molecular level. This is a remarkable admission. We have been routinely inducing unconsciousness in surgical patients since the 1840s, and we do not know exactly how it works. What we do know is that anesthetics interact with proteins in neural membranes. Roger Penrose and Stuart Hameroff have argued specifically that anesthetics act on microtubules, supporting their theory that microtubules are involved in consciousness. The idea is that if anesthetics disrupt quantum processing in microtubules, this would explain why they eliminate consciousness without simply killing the neurons. The neurons survive anesthesia. Their electrical activity continues to some extent. What disappears is the integrative experience that we call being aware. If this is correct, a quantum simulation of microtubule dynamics under anesthetic exposure would directly illuminate the physical mechanism of consciousness. This hypothesis is testable, and the fact that it is testable is what makes quantum simulation of biological systems scientifically valuable. Competing theories of consciousness make different predictions about what a sufficiently detailed simulation would show. A simulation capable of faithfully reproducing the molecular dynamics of neural tissue would provide a platform for discriminating between these theories in a way that purely conceptual analysis cannot. We would be running experiments on the physical substrate of mind. To simulate these dynamics requires not just quantum hardware, but also advances in how quantum computers are programmed and controlled. Quantum algorithms for simulating molecular dynamics, called variational quantum eigensolvers and quantum phase estimation algorithms, have been developed and tested on small systems. They work. The results match what we expect from classical quantum chemistry calculations. The limitation is that the systems they can handle are still small, on the order of dozens of electrons in a molecular orbital structure. Scaling these algorithms to handle proteins with thousands of atoms requires hardware that does not yet exist at the required scale. But the algorithms themselves are ready. There is also a growing body of work on quantum machine learning algorithms specifically relevant to the problem of decoding neural activity. Classical machine learning has been successfully applied to decode motor intentions from neural recordings, enabling basic brain-computer interfaces. But classical decoders face fundamental limitations in the quality and speed of decoding complex cognitive states. Quantum machine learning algorithms can, in principle, find patterns in high-dimensional data that classical algorithms cannot detect efficiently. Applying these to the high-dimensional probability distributions of neural activity could dramatically improve the quality and latency of brain-computer interface decoding, even before a full brain simulation is achieved. The interaction between quantum computing and neuroscience is therefore not a single monolithic project. It is a collection of converging research programs, each addressing a specific aspect of the grand challenge, and each capable of delivering scientifically and medically meaningful results at different points along the development timeline. Error correction research advances the stability of quantum hardware. Quantum chemistry algorithms advance our ability to simulate molecular-level neural processes. Quantum machine learning algorithms advance the quality of brain-computer interfaces. And foundational research into quantum effects in biological systems advances our understanding of whether and how the brain itself uses quantum mechanics. All of these programs are active today. There is one more dimension of this topic that deserves careful attention. It concerns the distinction between artificial intelligence and what might be called neuromorphic quantum intelligence. Modern artificial intelligence systems, particularly the large language models that have become prominent in public discourse, are trained by exposure to data. They learn statistical patterns in text, images, or other data types, and generate outputs that reflect those patterns. They are extraordinarily sophisticated pattern matchers. They can produce fluent text, recognize images, write code, and generate plans. They are also brittle in ways that biological intelligence is not. They fail at problems that require genuine reasoning outside their training distribution. They lack the flexible generalization that characterizes human cognition. The reason for this brittleness is architectural. These systems are trained to minimize a loss function over a training dataset. Everything they know is derived from what they have seen. They have no way to engage with novel problems by reasoning from first principles, because first principles require access to the underlying physics of the world, which is quantum mechanical, and which cannot be captured in a dataset of text and images. A quantum simulation of neural dynamics is a different kind of thing entirely. It is not trained on data. It is built from physics. Its behavior emerges from the same equations that govern the behavior of the actual biological system it is modeled on. If the model is sufficiently accurate, it does not just replicate what we know. It can explore what we do not know because it is running the same physics that the brain runs. It can discover things that are not in any dataset because it is operating from the same first principles as nature. An artificial intelligence system can tell you things that are consistent with the patterns in its training data. A quantum neurosimulation can do experiments. It can be given novel conditions that were never part of any training dataset and generate responses by following the physics, not by extrapolating from examples. This is the difference between an encyclopedia and a laboratory. The history of science is full of examples where a powerful new instrument opened a window onto phenomena that were previously invisible. The telescope revealed the moons of Jupiter and the craters of the moon, overturning the Aristotelian picture of perfect celestial spheres. The microscope revealed the cellular structure of living tissue, overturning the humoral theory of disease. The functional MRI scanner revealed the large-scale patterns of neural activity during cognition, overturning the localizationist view that individual cognitive functions were confined to small, fixed brain regions. A quantum brain simulator would be an instrument of this same category. It would reveal aspects of neural dynamics that are currently invisible to all existing tools. The most important findings are always the ones we could not have predicted in advance, because if we could have predicted them, we would not have needed the instrument. The question of what the brain is actually optimizing for is one that the mainstream discussion of quantum computing almost always skips over. Classical computers are designed to optimize for a specific output, the correct answer to a well-defined problem. You give the machine a question, it computes the answer. This is enormously useful, But it is not what the brain is doing. The brain did not evolve to solve calculus problems. It evolved to keep an organism alive in a complex, uncertain, rapidly changing environment. That goal requires a completely different computational strategy. An organism facing a predator cannot afford to wait for a guaranteed optimal solution. It needs a fast, good enough decision made under severe uncertainty, with incomplete information, under time pressure, while simultaneously managing 100 other physiological processes. The brain's architecture reflects these constraints at every level. One of the most striking features of neural processing that reflects this imperative is predictive coding. The prevailing model of visual processing used to be feed-forward. Light hits the retina, signals propagate through a hierarchy of visual areas, and somewhere at the top of the hierarchy, an object is recognized. This model is wrong, or at least deeply incomplete. The brain's visual system is not primarily a bottom-up signal processor. It is a prediction machine. At every level of the visual hierarchy, higher brain areas are sending predictions downward, anticipating what the lower areas are about to signal based on prior experience and current context. The lower areas compare the incoming sensory signal with the prediction from above. If the signal matches the prediction, almost nothing happens. If the signal deviates from the prediction, a prediction error is sent upward. Only the unexpected, the things that violate the brain's model of the world, generate strong neural activity. The brain is running a constant simulation of its environment and updating that. Simulation based on deviations from prediction. This predictive coding framework means that the brain is not primarily processing reality. It is constructing a model of reality, verifying it against incoming data, and updating it when the model is wrong. The subjective experience of perceiving the world is largely a simulation generated by the brain's internal model, with sensory data serving mainly as error signals that correct the simulation when it goes off course. This is a radical reconceptualization of perception, a quantum simulation of the brain that accurately models neurodynamics, must therefore model not just the passive processing of sensory signals, but the active generation of predictions. It must model how prior experience shapes the expectations that are projected downward from higher to lower brain areas. It must model how prediction errors propagate upward and update the internal model. This is a much richer computational picture than the simple feed-forward circuit diagrams that dominated neuroscience for decades. And it is a picture that maps naturally onto the quantum mechanical framework, the Bayesian brain hypothesis, which generalizes predictive coding to a full statistical framework, argues that the brain is essentially implementing Bayesian inference. Given a prior distribution over possible states of the world and incoming sensory data, the brain computes the posterior distribution, the updated probability distribution over world states given the data. This is optimal inference under uncertainty. It is also computationally expensive for a classical system because exact Bayesian inference in high-dimensional spaces is exponentially hard. One intriguing hypothesis is that the brain implements approximate Bayesian inference using quantum sampling. Quantum systems naturally sample from probability distributions that are defined by their wave functions. If the brain's quantum dynamics effectively implement a quantum sampling process over the space of possible world states, the result would be approximate Bayesian inference at biological speed and efficiency. This would explain both the brain's remarkable efficiency and its characteristic pattern of errors and biases in perception and decision-making. This hypothesis makes a specific prediction. The brain's perceptual and cognitive errors should follow the statistics of quantum sampling from a Bayesian posterior, rather than the statistics of deterministic classical approximation. If the hypothesis is correct, It would mean that the brain's architecture is not just efficient in an engineering sense. It is optimal in a physical sense. It is the most efficient possible way to implement probabilistic inference given the physical resources available to it. Evolution, which is itself a process of optimizing fitness over millions of generations, may have discovered the quantum mechanical foundation of optimal inference and built it into every neuron. The idea also connects to a broader theme that has emerged from the study of quantum biology, that living systems are not just subject to the laws of physics, but have evolved to exploit them in sophisticated ways. Photosynthesis uses quantum coherence to route energy efficiently from light harvesting complexes to the reaction center. The navigation of migratory birds may exploit quantum entanglement in cryptochrome proteins to sense the direction of the Earth's magnetic field. The sense of smell may involve quantum tunneling of electrons in odorant molecules. In each of these cases, evolution has not been limited to the crude material of classical mechanics. It has dug down to the quantum level and found mechanisms of efficiency and sensitivity that classical processes cannot match. The brain, as the most complex biological organ and the one subject to the most intense selection pressure, should be expected to have exploited quantum mechanics more extensively and more sophisticatedly than any other biological system. The evidence is accumulating that it has. There is a deeper question lurking underneath all of this, one that cannot be deferred indefinitely as the technology matures. It is the question of consciousness itself. If we build a quantum simulation of a human brain that accurately replicates the energy states, the protein dynamics, the firing patterns, and the quantum fluctuations of biological neural activity, what exactly have we built? Have we built a model that calculates data, or have we built something that experiences the calculation? This is what philosopher David Chalmers calls the hard problem of consciousness. The easy problems of consciousness, in his terminology, are the ones that can be solved by good enough neuroscience. We can, in principle, explain how the eye encodes visual information, how the hippocampus consolidates memory, how the prefrontal cortex evaluates decisions. These are difficult engineering problems, but they are, in kind, the same as any other scientific problem. The hard problem is different. It is the question of why any of that feels like something. Why does the specific pattern of activity associated with seeing red produce the subjective experience of redness? No equation in current physics answers this. Quantum mechanics offers an intriguing new angle on this problem. Integrated information theory, proposed by neuroscientist Giulio Tononi, argues that consciousness arises from the degree to which a system is irreducible. A conscious system is one where the whole is more than the sum of its parts, where the interactions between components generate information that cannot be decomposed into the sum of each component's individual contribution. The measure of this irreducibility is called phi. In a classical computer, components are separable. You can isolate one processor from another. Information flows between them, but each component is, in principle, describable independently. In a quantum computer, entangled qubits are not separable. They form a unified wave function. You cannot describe the state of one qubit without describing the state of the whole entangled system. This physical unity corresponds to a high degree of irreducibility. If integrated information theory is correct, an entangled quantum system would naturally tend to exhibit a non-zero value of phi. Whether this is sufficient for consciousness remains unknown, but it is a more credible substrate for integrated information than anything classical computing provides. If this line of reasoning holds, then a sufficiently detailed quantum simulation of a brain might not merely model consciousness, it might instantiate it, and that possibility draws a boundary we have never had to draw before. If we create a quantum simulation of a human mind for the purpose of testing a drug, and that simulation generates signals consistent with pain, are we ethically obligated to treat those signals as meaningful? In a classical simulation, dismissing them as mere code is defensible. The classical computer is printing the word pain. The word pain and the state of pain are clearly different things. In a quantum simulation that is physically isomorphic to a brain in distress, the distinction becomes less clear. The physical state of the machine may be, in some morally relevant sense, the same as the physical state of a person suffering. This is not a distant philosophical puzzle. It is an engineering deadline. As quantum simulations become more detailed and more biologically accurate, the scientific community will need a framework for evaluating the moral status of simulated systems before the technology makes the question urgent. We do not currently have such a framework. We do not even have consensus on what features of a physical system give rise to experience in the first place. The role of in shaping neural dynamics is so pervasive and so fundamental that no simulation of cognition can be considered complete without modeling it accurately. The brain does not run at a fixed operating point. Its computational properties change continuously depending on the levels of neuromodulatory chemicals like dopamine, serotonin, norepinephrine, and acetylcholine. These chemicals are released by small clusters of neurons in the brainstem and diffuse across large volumes of brain tissue, modulating the responses of billions of neurons simultaneously. These chemicals set the tone of neural processing. They determine whether the brain is in a mode of focused exploitation of known solutions or open exploration of novel possibilities. They regulate the speed of learning, the threshold for action, the salience of reward, and the capacity for sustained attention. In terms of their physical mechanism, neuromodulators work by binding to receptors on the surface of neurons and triggering intracellular signaling cascades. Every step of this cascade involves quantum mechanical processes in proteins. The binding of a dopamine molecule to its receptor involves quantum mechanical recognition between molecular surfaces. The activation of the downstream signaling enzyme involves a conformational change driven by electron transfer. A quantum simulation of neuromodulatory signaling would be a scientific milestone in its own right. Many of the most important drugs in psychiatry work precisely by altering neuromodulatory signaling. Antidepressants modify serotonin levels. Antipsychotics target dopamine receptors. Stimulants enhance norepinephrine and dopamine signaling. We design these drugs by a combination of informed guesswork and clinical trial and error. A quantum simulation capable of modeling the full molecular cascade of neuromodulatory signaling would transform psychiatric drug development from art to engineering. The dopaminergic system, in particular, is intimately connected to some of the deepest questions in neuroscience and behavioral science. Dopamine is often described simply as the brain's pleasure chemical. Contemporary neuroscience understands dopamine as primarily a reward prediction error signal. Dopamine neurons fire not when something good happens, but when something better than expected happens. If the brain predicts that pressing a lever will yield no reward and a reward appears, dopamine neurons fire strongly. If the brain predicts a reward and the reward arrives exactly as predicted, dopamine neurons barely fire. If the brain predicts a reward and it does not appear, dopamine neurons are suppressed below baseline. This reward prediction error signal is what drives learning. It is the engine of habit formation, skill acquisition, and motivated behavior. The quantum mechanical sensitivity of the molecular machinery driving dopaminergic neurons may be part of what allows this system to detect such fine-grained differences in the timing and magnitude of reward. A quantum simulation of the dopaminergic system would allow researchers to test this hypothesis directly. Does the quantum noise in the molecular machinery create variability in the reward prediction error signal that corresponds to the observed variability in human learning? Does disrupting specific quantum mechanical steps in the signaling cascade alter the precision of reward learning in ways that match the symptoms of conditions like addiction, depression, and schizophrenia? These are empirically meaningful questions. Answering them would advance our understanding of the neuroscience of mental illness, far beyond what current experimental techniques can achieve. There is a feedback loop between neuromodulation and the quantum dynamics of neural tissue that deserves special attention. Neuromodulators alter the expression of genes inside neurons, changing which proteins the neuron produces over timescales of hours and days. These changes in protein composition alter the quantum mechanical properties of the neuron's molecular machinery. The neuron that exists tomorrow is physically different from the neuron that exists today because the pattern of activity it experienced today has changed its gene expression. This is the molecular substrate of memory. Long-term memory storage involves the synthesis of new proteins at specific synapses, proteins that structurally modify the synaptic connection to make it more or less efficient. A synaptic connection that carries a significant pattern of correlated activity is marked for structural modification, while neighboring connections carrying random or uncorrelated activity are not. This selectivity, the ability of the synapse to distinguish meaningful correlated activity from background noise, may depend on quantum mechanical processes that classical models cannot capture. The NMDA receptor, a specific type of glutamate receptor, plays a central role in this process. NMDA receptors act as coincidence detectors. They open only when two conditions are met simultaneously. The presynaptic neuron must release glutamate, and the postsynaptic neuron must already be depolarized by other inputs. This and gate property is what allows the synapse to detect correlated activity. When the NMDA receptor opens, it allows calcium to flow into the postsynaptic spine. The calcium signal triggers the intracellular cascade that ultimately leads to protein synthesis and synaptic strengthening. The opening of the NMDA receptor requires not just the binding of glutamate, but also the removal of a magnesium ion that normally blocks the channel. This magnesium block is relieved only when the postsynaptic membrane is sufficiently depolarized. The timing of this event, the precise moment at which depolarization is sufficient to relieve the magnesium block while glutamate is still bound, is determined by the quantum mechanical dynamics of ion binding and channel conformation. A quantum simulation of NMDA receptor function would generate predictions about the precision of synaptic coincidence detection that could be tested against electrophysiological measurements. It would also generate predictions about how specific mutations in the NMDA receptor, many of which are associated with schizophrenia and autism spectrum disorder, alter the coincidence detection process. The difference between a statistical model and a physical simulation is not just mathematical. It is the difference between describing what happens and understanding why it happens. Understanding why things happen at the quantum mechanical level is what allows us to make predictions about novel situations, including the effects of drugs that have never been synthesized, mutations that have never been observed in a patient, or environmental conditions that have never been studied experimentally. There is one more thread worth pulling because it connects the physics of quantum computation to a question that is more personal than any of the others we have discussed. That question is about sleep. Every night, the human brain undergoes a process of dramatic reorganization. Synaptic connections that were active during the day are selectively pruned or strengthened. New proteins are synthesized at specific synapses, waste products that accumulate during waking activity, including misfolded proteins like the beta amyloid associated with Alzheimer's disease, are cleared from the brain tissue through a system of fluid channels that is more active during sleep than during wakefulness. Memory traces formed during the day are consolidated through a process of replay in which the patterns of activity that encoded a new experience are reactivated. During slow wave and rapid eye movement sleep, the neural dynamics of sleep are as complex and as important as the neural dynamics of waking cognition, and they are as poorly understood. The molecular mechanisms by which specific memories are selected for consolidation during sleep and others are allowed to decay are unknown in detail. The relationship between the oscillatory rhythms of sleeping brain activity, the slow waves of non-REM sleep, and the rapid oscillations of REM, and the molecular processes of synaptic modification that occur during these rhythms is known only in broad strokes. A quantum simulation of neural dynamics during sleep would be one of the most scientifically valuable experiments that could be run on such a platform. Sleep disturbances are associated with virtually every psychiatric and neurological condition. Chronic sleep deprivation is one of the most reliable ways to impair cognition and increase the risk of mental illness. The accumulation of amyloid during periods of insufficient sleep is one of the most compelling risk factors for Alzheimer's disease. Understanding the quantum mechanical details of what the brain is doing during sleep and why disrupting sleep disrupts health so severely could transform our ability to treat a vast range of conditions. It would also advance our understanding of consciousness from the negative space. Consciousness, whatever it is absent during dreamless sleep. The brain is still active during this phase, still running its molecular machinery, still clearing waste and consolidating memory, but the lights are off. Something that is present during waking and during REM dreaming is absent during dreamless sleep. If quantum simulation can identify what physical process, what pattern of quantum dynamics is present in the conscious state and absent in the dreamless sleep state, that identification would be among the most significant findings in the history of neuroscience. This is a concrete experimental question. It is not asking the abstract philosophical question of what consciousness is. It is asking the physically precise question of what changes between the waking state and the dreamless sleep state that could account for the difference in conscious experience. A quantum simulation that accurately models both states and identifies the physical difference would provide the strongest empirical constraint ever placed on theories of consciousness. The circadian rhythm that governs the sleep-wake cycle is itself regulated by molecular processes that are explicitly quantum mechanical. The core of the circadian clock is a transcription-translation feedback loop involving proteins called clock, BMAL1, PER, and CRY. The binding of these proteins to DNA and to each other, which drives the 24-hour oscillation of the clock, involves the same quantum mechanical molecular recognition that governs every protein-protein and protein-DNA interaction. Mutations in circadian clock genes cause severe circadian rhythm disorders, and these disorders are associated with increased risk of cancer, metabolic disease, psychiatric illness, and neurodegenerative disease. A quantum simulation of the molecular circadian clock would generate predictions about how specific mutations alter the timing and robustness of the oscillation. These predictions could be tested in cell cultures carrying the specific mutations using fluorescent reporters of clock gene expression to track the oscillation in real time. The accuracy of the circadian clock is itself a remarkable physical phenomenon. A molecular oscillator involving a small number of protein molecules manages to keep time with a precision that the most accurate classical mechanical clocks took decades of engineering refinement to achieve. Whether quantum coherence plays a role in the noise suppression that makes this possible is an open question that quantum simulation could address. Every biological system we look at closely enough reveals quantum mechanical features that contribute to its function. The precision, efficiency, and flexibility of biological systems cannot be fully explained by classical chemistry. Quantum mechanics is the missing layer of physical description, and its absence from our computational models of biology, including the brain, is responsible for the gap between what our models predict and what our experiments observe. The closing of that gap is what the quantum brain simulation program is about. The coming years will bring more results from quantum hardware platforms, more discoveries from quantum biology experiments, more demonstrations of quantum advantage for specific molecular simulation problems, and more refinements of the theoretical frameworks connecting quantum mechanics to neuroscience. Each of these developments will be a step toward the long-horizon goal of a real-time, physically faithful simulation of the human brain. When that goal is achieved, the scientific consequences will be profound. We will understand the brain as a physical system in a way that has never been possible before. We will be able to treat neurological and psychiatric diseases with a precision that is currently unimaginable. We will have brain-computer interfaces that are seamless extensions of biological cognition rather than clumsy prosthetics. And we will have confronted with empirical tools rather than purely philosophical ones the deepest question that science has yet approached. The question of what makes physical matter into an experiencing mind. We are living in a remarkable moment. The combination of quantum hardware, quantum algorithms, experimental techniques in quantum biology, and theoretical advances in neuroscience has created an opportunity that did not exist a generation ago. The scientific and technological pieces are coming together in a way that makes previously impossible investigations possible. The questions have always been there. The tools are becoming real. The energy gap between the brain and the supercomputer was always a signal. It was nature's way of telling us that we were looking in the wrong direction. Quantum mechanics is the right direction. The brain knew this all along because it had no choice. It was built from physics, and physics is quantum. We are finally catching up. What is still genuinely unknown is where the quantum signature in the brain actually lives. The evidence for quantum processes in microtubules is suggestive but not yet conclusive, demonstrating unambiguously that quantum coherence persists long enough in a warm biological environment to influence neural computation requires measurements at the boundary of what current instrumentation can achieve. This is not a reason for skepticism. It is a reason for more careful science. What is also still unknown is how consciousness relates to the physical substrate. Integrated information theory is one framework. Global workspace theory is another. Predictive processing models offer yet another angle. None of them is definitively proven. None of them is definitively wrong. The hard problem remains hard, but the tools we are building to simulate the brain will generate data that bears directly on these questions in ways that pure philosophical analysis cannot. The gap between classical computing and the brain has been visible for decades. We measured it in watts, in synapses, in seconds of simulation time. We diagnosed it as a fundamental incompatibility between the physics of silicon and the physics of biology. Now we are building, step by step, the tools to bridge it. The qubits are getting cleaner, the error rates are falling, the algorithms are becoming more sophisticated. The biology is revealing its quantum architecture more clearly with every experiment. The bridge is under construction. The universe is made of quantum mechanics. The brain is made of the universe. And we are finally building computers made of the same physics as both. If you want to follow what comes next, because what comes next in this research is going to be worth watching, consider subscribing. The results emerging from quantum hardware, the discoveries from quantum biology, the advances in brain-computer interfaces. This is an area where the news from the laboratory genuinely changes what is possible to think. That is a rare thing, and it is happening now. To fully appreciate the magnitude of what quantum brain simulation represents, it helps to trace the longer arc of how scientists have understood the relationship between computation and the physical world. For most of the 20th century, the dominant assumption was simple. The brain is a computer, and computers are, in principle, capable of simulating the brain. This view was enormously productive. It gave birth to cognitive science, to computational neuroscience, 2 decades of research that produced genuine insights into how the brain processes sensory information, stores memory, and generates behavior. But the assumption concealed a flaw that grew more visible with each passing decade. The computer that scientists had in mind when they invoked this analogy was the classical Turing machine, a theoretical device that processes information by reading and writing symbols on a tape according to a finite set of rules. This device was designed to capture the essence of mathematical calculation, not the essence of physical reality. It was never claimed to be a model of the physical world. It was a model of logic. When scientists assumed that the brain is a Turing machine, they were assuming that cognition is fundamentally logical rather than physical. That assumption turns out to be wrong in ways that matter enormously. Cognition is not logical computation running on top of physics. Cognition is physics. It is a specific pattern of physical dynamics that arises from the quantum mechanical behavior of biological matter. Understanding it requires not a better logical model, but a better physical model. This is the conceptual shift that the quantum brain simulation program represents. It is a shift from modeling the brain as a logical system to studying the brain as a physical system. And the tools required for that shift are quantum mechanical by necessity, because the relevant physics is quantum mechanical at its foundation. Consider what this means for the interpretation of memory. Classical computational models of memory treat it as a data storage and retrieval problem. Information is encoded in binary patterns, stored at specific addresses, and retrieved by accessing those addresses. This model captures something real about how the brain behaves at a coarse level, but it misses the physical reality of how memory actually works. At the molecular level, memory is not stored as a pattern of bits. It is stored as a physical modification of synaptic connections, a change in the molecular structure of the protein complexes that constitute the synapse. The information is not written into a register. It is embedded in the geometry of macromolecules. Reading that information out is not a digital lookup operation. It is a physical process in which the geometry of the molecular machinery influences the dynamics of neural activity. The quantum mechanical details of this molecular geometry are not incidental. They are the mechanism. The specific conformational state of the proteins at a modified synapse determines the efficiency with which that synapse transmits signals. That efficiency depends on the positions and quantum states of electrons within the protein structure. A classical description of the protein can capture its average behavior, but it averages away the quantum mechanical details that determine the precision and fidelity of the synaptic transmission. This is why quantum simulation is not just a quantitative improvement over classical simulation. It is a qualitative step into a different level of physical description. the level at which the actual mechanism of cognition operates. The implications extend to our understanding of learning itself. Learning, at the neural level, is the process by which experience modifies the strength of synaptic connections. The rules governing this modification, known as synaptic plasticity, determine what the brain learns and how quickly. The most fundamental form of synaptic plasticity follows a principle articulated by psychologist Donald Hebb in 1949. Neurons that fire together, wire together. Synapses connecting neurons that are consistently active at the same time are strengthened. Synapses connecting neurons that rarely coincide in their activity are weakened. But the Hebbian principle is a description, not an explanation. It tells us what the rule of synaptic modification is, not how the synapse detects coincident activity and implements the modification. The molecular explanation involves the NMDA receptor acting as a coincidence detector, as described earlier. But the NMDA receptor's ability to detect coincident activity depends on the precise timing of ion-binding events, and that timing is governed by quantum mechanical dynamics that classical models can only approximate statistically. If the quantum mechanical details of NMDA receptor function differ between individuals due to genetic variation, those differences could manifest as differences in the precision and efficiency of learning. This could explain some of the remarkable variation in learning speed and memory consolidation that is observed between individuals. Even in populations without any diagnosed neurological disorder, a quantum simulation capable of modeling NMDA receptor function across different molecular configurations would allow researchers to predict how specific genetic variants affect learning at the molecular level, and to test those predictions against behavioral measurements in humans carrying those variants. The relationship between quantum mechanics and neurodevelopment is another area that has received insufficient attention in the mainstream discussion. The brain does not arrive fully formed. It develops over decades, with the most dramatic structural changes occurring during prenatal development and early childhood, but with significant ongoing reorganization continuing through adolescence and into early adulthood. The process by which neurons find their targets during development, migrating to their correct positions and extending axons that navigate precisely to the correct synaptic partners across distances that are enormous, relative to the size of the growing neuron, is one of the most remarkable feats of biological self-organization known to science. This navigation is guided by molecular cues, gradients of signaling molecules that the growth cone of the extending axon detects and responds to by altering its direction of growth. The molecular machinery by which the growth cone detects these gradients and translates them into directional movement involves the same quantum mechanical protein dynamics that underlie synaptic transmission in the adult brain. Mutations that disrupt these quantum mechanical steps can derail neural development in ways that produce neurodevelopmental conditions including autism spectrum disorder and schizophrenia. A quantum simulation of the molecular machinery of axon guidance would allow researchers to predict how specific mutations alter the development of neural circuits, and to test those predictions against developmental imaging data from individuals carrying the mutations. This could provide an entirely new angle on the neurobiology of neurodevelopmental conditions, connecting the molecular genetics to the circuit-level abnormalities that produce behavioral symptoms through the quantum mechanical mechanism that links them. The question of how the brain generates creativity also looks different through the lens of quantum mechanics. Classical computational models of creativity typically invoke some combination of random search, through a space of possible ideas and selection of those that meet specified criteria. This model is uninspiring not just aesthetically but scientifically, because it does not capture what actually happens in the brain during creative thought. Neuroimaging studies of creative cognition have identified a network of brain regions, including the default mode network, the executive control network, and the salience network, that interact in distinctive ways during creative problem solving. The default mode network, which is active during mind-wandering and spontaneous thought, provides a rich, generative substrate. The executive control network provides directed evaluation and selection. The salience network coordinates the interaction between these two. The dynamics of this three-way interaction are not captured by simple, random search models. What quantum mechanics offers to the understanding of creativity is a different picture of how the brain generates novel combinations of ideas. If the brain's quantum dynamics implement something like quantum sampling from a Bayesian posterior over possible world models, then the process of creative insight could be understood as the brain's quantum sampling process, arriving at low probability but high value states that are inaccessible to the more directed, high probability sampling that characterizes routine thought. Insight would be, on this view, a quantum fluctuation that reaches an unexpected region of the space of possible ideas, a region that rational directed search would not have explored. This is testable. If creative insights correspond to quantum fluctuations in the brain's sampling process, they should show specific signatures in the dynamics of neural activity that precede the moment of insight. These signatures would be distinct from the neural dynamics of routine problem solving, and they would reflect the quantum mechanical properties of the underlying neural sampling process. A quantum simulation of the neural dynamics of creative cognition would generate specific predictions about these signatures that could be tested against higher resolution neural recordings in subjects performing creative tasks. The practical applications of such an understanding would extend far beyond neuroscience. Creativity is central to scientific discovery, artistic production, technological innovation, and cultural evolution. Understanding the physical mechanism of creativity at the quantum mechanical level would provide a foundation for designing interventions, whether pharmacological, neurological, or educational, that enhance creative capacity while preserving the richness and unpredictability that make human creativity valuable. Another area that deserves attention in this context is the neuroscience of expertise. When an individual develops deep expertise in a domain, whether as a chess grandmaster, a skilled musician, a master surgeon, or an expert physicist, the neural changes that accompany the acquisition of expertise are extensive and deep. The white matter tracks connecting relevant brain regions become more organized and efficient. The gray matter volume of relevant cortical regions changes. The patterns of neural activity during expert performance become both more efficient and more precise. These changes reflect modifications at the synaptic level that have accumulated over thousands of hours of practice. The molecular details of these modifications, the specific quantum mechanical changes in the protein structures of the synapses that encode expert knowledge, are essentially unknown. Classical neuroscience can describe the statistical patterns of neural activity in experts and compare them to novices, but classical models cannot explain why. Specific patterns of practice drive specific molecular modifications that produce specific improvements in PERFORMANCEA, quantum simulation of the molecular dynamics of synaptic modification. During skill, learning would allow researchers to model the relationship between specific patterns of neural activity and specific molecular changes at the This could reveal the quantum mechanical rules that govern the conversion of experience into durable structural modifications of neural circuits. Understanding these rules would transform the science of learning and education, providing a physical foundation for designing, training, protocols that maximize the efficiency with which practice translates into lasting neural modification. The connection between quantum mechanics and aging is another dimension of the story that deserves more attention than it typically receives. The brain ages, and the aging process involves progressive changes in the molecular machinery of neural function that reduce cognitive performance, increase the risk of neurodegenerative disease, and eventually compromise the ability of the brain to maintain its own structural integrity. At the molecular level, aging involves the accumulation of oxidative damage to proteins, lipids, and nucleic acids, the progressive dysfunction of protein quality control systems that normally identify and clear misfolded proteins, the shortening of telomeres in dividing support cells, and the emergence of chronic low-level inflammation in neural tissue. All of these processes involve quantum mechanical dynamics. Oxidative damage occurs when reactive oxygen species, which are quantum mechanical in their electronic structure, react with biological molecules and alter their quantum electronic states in ways that disrupt their function. Protein quality control systems rely on the same quantum mechanical recognition processes that underlie all protein-protein interactions. The emergence of inflammation reflects changes in the quantum mechanical signaling dynamics of immune cells in the brain. A quantum simulation of the aging brain would allow researchers to model how these molecular changes accumulate over time and how they interact to produce the cognitive changes associated with aging. It would allow the testing of interventions designed to slow specific quantum mechanical processes of molecular deterioration, providing a physically grounded basis for the development of anti-aging strategies for the nervous system. The potential applications in the treatment of age-related cognitive decline represent one of the largest unmet medical needs in the developed world. As populations age, the burden of Alzheimer's disease, vascular dementia, and other age-related cognitive conditions is growing rapidly. The economic cost of dementia care globally is measured in trillions of dollars annually. A physically grounded understanding of the quantum mechanical processes that drive neural aging could make the development of effective, preventive, and therapeutic interventions not just possible, but tractable. Looking further into the future, the convergence of quantum computing and neuroscience raises questions about the ultimate limits of cognitive enhancement. The brain, as it exists today, is the product of billions of years of evolutionary optimization under the constraints of biological implementation. Evolution found quantum mechanical solutions to the problems of efficient computation, robust memory, and flexible generalization. But evolution is constrained by what is biologically feasible. It cannot implement arbitrary physical systems. It can only select from the variation that arises from genetic mutation and recombination. A technology capable of designing quantum mechanical systems from first principles, unconstrained by the limitations of biological implementation, could, in principle, implement cognitive architectures that exploit quantum mechanics more efficiently than the brain does. This is the long-horizon vision that animates some of the researchers working at the intersection of quantum computing and neuroscience. Not just simulating the brain, but understanding the brain well enough to build cognitive systems that improve on it. This is a distant goal, and the path to it is uncertain. But it is not obviously impossible. The brain is a remarkable physical system, but it is not a perfect one. It is subject to biases, blind spots, and failure modes that are the legacies of its evolutionary history. A quantum mechanical cognitive architecture designed from first principles, rather than evolved under biological constraints, might avoid some of these failure modes while preserving the core capabilities that make human cognition valuable. The scientific program required to reach this goal would be among the most ambitious in the history of technology. It would require a complete physical understanding of how the brain implements quantum mechanical computation, the development of quantum hardware capable of implementing arbitrary quantum Hamiltonians at the required scale, the development of theoretical frameworks capable of connecting quantum physical dynamics to cognitive-level phenomena, and the resolution of deep ethical questions about the moral status of artificial cognitive systems. None of these challenges is trivial. All of them are tractable in principle. The researchers working on quantum brain simulation are not motivated by this distant vision alone. They are motivated by the immediate scientific questions that their work addresses. How do proteins fold? How do ion channels open and close? How do synapses transmit signals with quantum mechanical precision? How does the brain maintain its remarkable energy efficiency? These are foundational questions in biology that quantum simulation is positioned to answer in the near term, with implications that extend across medicine, biotechnology, and fundamental science. The long horizon vision, a physical understanding of the quantum mechanical basis of cognition, is the destination that gives direction to the journey. But the journey itself, the step-by-step process of building quantum hardware, developing quantum algorithms, discovering quantum effects in biological systems, and building bridges between quantum physics and neuroscience, is scientifically valuable at every stage. We are at the beginning of that journey. The first steps have already been taken. The Willow chip demonstrated that quantum error correction can be engineered to work. Quantum biology has established that quantum coherence plays functional roles in photosynthesis, bird navigation, and possibly neurodynamics. Quantum algorithms for molecular simulation are running on existing hardware and producing results that match classical quantum chemistry for small systems. The theoretical frameworks connecting quantum mechanics to neuroscience are becoming more precise and more testable. The pace of progress in quantum hardware has consistently surprised the experts who've predicted it. A decade ago, the idea that a quantum processor with hundreds of logical qubits and sub-threshold error rates would exist in 2024 was optimistic to the point of skepticism. The Willow result changed that assessment. The next decade of progress is likely to be equally surprising in the direction of faster than expected advancement. What we know with confidence is that the approach is correct. The physics is right. The brain is a quantum system, and the inadequacy of classical simulation reflects the fundamental incompatibility between classical mechanics and quantum reality. The tools being built to bridge that incompatibility are advancing along a trajectory that makes the long-term goals achievable, and the questions those tools will help answer about cognition, about consciousness, about the physical basis of mind, are among the most important questions that science has ever pursued. The story of quantum brain simulation is, at its deepest level, a story about the relationship between physics and meaning. For most of the history of science, physics and meaning were studied separately. Physics asked what the world is made of and how it behaves. The study of mind asked what experience is and how it arises. These two inquiries operated in parallel, occasionally intersecting but never fully converging. Quantum brain simulation is where they converge. It subjects the physical substrate of experience to the same rigorous experimental and theoretical treatment that physics has applied to atoms and particles and fields. It brings the question of consciousness into the domain of experiment, where it can be addressed with precision rather than speculation. It makes the hardest question in philosophy, the question of why physical processes give rise to subjective experience, into a scientific question that can be investigated with instruments and measurements. Whether quantum simulation will ultimately answer that question remains to be seen. The hard problem of consciousness is hard for reasons that run deep, but the tools being built to investigate the quantum dynamics of the brain will, at minimum, generate the most precise empirical data that has ever been brought to bear on the question. They will constrain the space of possible answers in ways that pure philosophical argument cannot. They will force the development of better theories, and better theories will generate better experiments, and the iterative process of scientific inquiry will advance. This is how science has always worked. It advances not by solving problems in single brilliant strokes, but by building better instruments, generating better data, developing better theories, and repeating the cycle with increasing precision. The quantum brain simulation program is the beginning of that cycle applied to the most important and most difficult problem in science, the physical basis of mind. We began this account with an energy gap, 20 watts against millions, a discrepancy so large that it forces a question about the nature of computation itself. We end it with a convergence, quantum physics meeting neuroscience, the physics of matter meeting the physics of mind, the tools of physical science meeting the deepest questions of human experience. The convergence is real. The physics supports it. The biology is consistent with it. The engineering is building toward it. What waits on the other side of that convergence, we cannot fully anticipate. But if the history of science is any guide, it will be stranger, more interesting, and more significant than anything we currently expect. The universe is more remarkable than our current frameworks can capture. The brain, which is the universe's most intricate attempt to understand itself, is no exception. We are finally building the tools to help it succeed. There is one final dimension of this story that brings everything together, and it has to do with the nature of scientific revolutions themselves. Every major advance in our understanding of nature has required abandoning an assumption, so deeply embedded in our thinking that we did not recognize it as an assumption at all. Newton's mechanics assumed that space and time were absolute, unchanging containers within which events took place. Special relativity showed that this assumption was wrong, that space and time are relative to the observer, that simultaneity is not absolute, that the universe is stranger than the comfortable geometry of everyday experience suggested. Classical thermodynamics assumed that the behavior of large numbers of molecules could be understood statistically, without reference to quantum mechanics, that the laws of heat and energy were independent of the quantum nature of matter. The discovery of quantum mechanics showed that this assumption was also wrong, that the correct statistical mechanics of matter at any temperature requires quantum mechanics, that classical statistical mechanics is an approximation valid only in regimes where quantum effects are small. The assumption underlying classical computational neuroscience is that the computations of the brain can be captured by classical information processing models, that the relevant physics is classical, that quantum mechanics is a detail of the implementation that does not change the computational character of what the brain does. The evidence accumulating from quantum biology and the physical argument from Feynman suggests that this assumption is also wrong. The brain's computations are not classical computations with quantum mechanics as an irrelevant implementation detail. They are quantum computations, processes in which the quantum mechanical behavior of electrons and ions and proteins is not a background constraint, but the active mechanism of cognition. The classical approximation captures some behaviors of the brain, the way that classical statistical mechanics captures some behaviors of matter, but it misses the quantum mechanical heart of the phenomenon. Recognizing this is the beginning of a new chapter in neuroscience, not the end of a long debate. The classical approach was not wasted. It built the foundation of knowledge, the detailed maps of brain anatomy, the careful recordings of neural activity, the behavioral experiments that revealed the structure of cognition, the genetic tools that identified the molecular players that make the quantum approach possible. The new approach does not discard that foundation. It builds on it. What changes is the physical framework within which the accumulated knowledge is interpreted and extended. The connector maps that classical neuroscience painstakingly assembled become blueprints for quantum simulation. The neural recordings that computational neuroscience analyzed statistically become validation targets for quantum models. The genetic variants that molecular neuroscience identified as risk factors for disease become parameters in quantum simulations of molecular dysfunction. Every piece of knowledge that neuroscience has accumulated becomes more valuable when it is placed within a quantum physical framework, because the quantum framework provides the causal mechanism that connects molecular structure to cellular function, to network dynamics, to cognition. Without the quantum framework, the connections are statistical and descriptive. With the quantum framework, they become mechanistic and predictive. This is the transformation that is underway. The path ahead is long and the challenges are genuine. Building quantum hardware that can simulate the full complexity of even a small neural circuit in real time will require years of engineering progress across multiple fronts. From qubit fabrication to error correction to quantum networking to algorithm design, the scientific challenges of validating quantum simulations of biological systems, of developing theoretical frameworks that connect quantum dynamics to cognitive level phenomena, of designing experiments that can test the predictions of quantum models against measurements in living tissue, are equally formidable, but the direction is clear. The physics points in one direction. The biology is consistent with that direction. The engineering is advancing along that direction. And the questions waiting at the destination, about the nature of memory and consciousness and experience and mind, are the questions that matter most. The tools are becoming real. The journey has begun. There is something fitting about the fact that the instrument required to understand the brain is a machine that operates by the same physical principles as the brain itself. When Galileo pointed a telescope at the sky, he was using an optical instrument to study optical phenomena. When biologists used microscopes to study cells, they were using light to study systems that use light for signaling. There has always been a deep connection between the instrument and the phenomenon it studies. The quantum computer is an instrument that uses quantum mechanics to study quantum mechanics, and the brain is a quantum mechanical system. The fit is as natural as it is profound. The quantum computers being built today are noisy, limited in scale, and demanding in their requirements for isolation and cooling. They bear the same relationship to the machines that will simulate neural circuits in real time that the earliest vacuum tube computers bore to modern processors. The principle is established. The engineering is being refined. The trajectory is clear, and the destination, a genuine physical understanding of the most remarkable object in the known universe. The human brain is within the horizon of what science can now see. Pay attention to what comes next. It will be worth it. The questions that have driven this account are not abstract. They are questions about what we are, how we work, why we experience the world the way we do, and what it would mean to truly understand that. Science has never been closer to having the tools to answer them. The bridge between quantum physics and the physical basis of mind is under construction, and the builders know where they are going.